for today.
So thank you so much, Naveen. I would like to first thank the organizers, Naveen and all of the other organizers for this phenomenal conference. You guys are very lucky here at KAUST. And this is my first time at KAUST. This is my first time in Saudi Arabia, and it's been really a pleasure here. Okay, so in the last 2 days, you have heard a lot of talks about the biology of personalized medicine. I'm going to shift gears right now, and I'm going to tell you about the chemistry of personalized medicine. So for a new field like personalized medicine, how do we fabricate the new materials that are going to be highly performant in this field? How do we do that? How do we fabricate new materials that are going to respond to the needs of, for example, gene therapy, or the needs of immuno-oncology, or the needs of biologics drug delivery? So these are all the questions that we try to answer as chemists. And in my research group in particular, what we work on is the molecule DNA. Now, you know DNA as the information molecule in our body, but what we're going to do today is we're going to take it out of its biological context completely. And we're going to use it as a molecular building block. So we're going to think of it as a molecule. We're going to use it as a molecule, as a building block. We're going to make materials from DNA. And then we're going to see whether these materials actually do respond to the needs and to the basically the requirements of these new areas of precision and personalized medicine. Okay, so I think that you're all familiar with drug delivery by now, and you also even saw this class of drug delivery vehicles before. To give you an example, essentially, we have this chemotherapeutic, doxorubicin, which is a cancer chemotherapeutic, but it has severe cardiac side effects. 30 years ago, the first generation of drug delivery vehicles was put together, and this is basically a lipid type of nanoparticle with the doxorubicin on the inside and with PEG chains, basically just polymer chains on the outside. And this very simple device was able to deliver doxorubicin without the cardiac side effects. And since then, the field of drug delivery has really taken off. This is 30 years ago till now, thousands of really very, very beautiful applications. And you saw some very, very nice ones in the last 2 days. However, we do have some barriers that we have to overcome in drug delivery. As an observation, there's something that we call the valley of death. Basically, what I'm talking about is there are thousands and thousands of new research publications and contributions that show us new drug delivery vehicles, but very few of them actually make it to the clinic. So the patients cannot actually benefit from these drug delivery vehicles. And so what I'm going to do is basically try to explain to you in general terms some of the barriers that the field is experiencing and how maybe DNA nanotechnology can actually help us with these barriers. Okay, the first thing is that for a long time we thought that if you have a drug delivery vehicle and you control the chemistry of that little container, that's fine. You just send it down the body and it will deliver. Well, it turns out that there are other parameters that influence how this is going to deliver, and they are size and shape. So size and shape determine everything. They determine how a drug delivery vehicle circulates in the body, how fast, whether it's cleared very quickly, what organ does it target, whether it enters diseased cells or not. All of this is actually determined by size and shape. Yet when you look at the clinically approved drug delivery vehicles, first of all, you see that they're mostly all spherical, even though there have been now a number of studies that have shown that non-spherical drug delivery vehicles are superior in many ways, but they're all spherical, and more importantly, they are not a single molecular weight, a single type of structure. They are basically a distribution of different sizes. And the FDA, of course, has a hard time approving mixtures, and more importantly, what we do not have is a true, what chemists call structure-activity relationship. So that is, can we take a single material, make a very large number of different shapes and structures, and ask the question, what do these materials do in the body? Okay? And at this point in time, there are not many or not any single materials where you can make a lot of nanomaterials from these and study these types of properties. Okay. Second problem. Personalized medicine, as you saw in the last 2 days, involves also biomarker detection. What I mean by that is that if you have, let's say, cancer, the cancer might actually have on the cancer cells, let's say, specific molecules, specific proteins that are really specific to the cancer cells and not to the normal cells. Okay? That's fantastic, because what you can now do is make a container, and the container is going to have on the outside these ligands. I don't know if I can point at them. Basically, these little ligands, okay? And these little ligands might actually bind to the proteins that are sitting on the surface and facilitate, basically, delivery. That's what it is. But it turns out that how the ligands are presented on the surface of a drug delivery vehicle matters, not just what the ligands are. And right now, most of the materials that we make have basically an isotropic or a complete distribution of the ligands on the outside, whereas this matters a lot. Matters so much that if you were kind of trying to imagine a world where you can create something like this, a nano container where you can place multiple different ligands on the outside in precisely different geometries, okay, then you might be able to start answering a lot of unanswered problems, and, for example, personalized vaccines, understanding receptor organization on cells, antivirals that also bind to viruses, because viruses display their antigens actually in a very well-defined geometrical way, etc. So basically, that kind of control over structure is really missing from nanomaterials made now for drug delivery. Now, I told you about proteins that are overexpressed on cancer cell surfaces. The other thing that is sometimes overexpressed and you want to target are genes and gene products like mRNA, microRNA. These are actually very important. They're nice biomarkers, they're personalized biomarkers, and there's a lot of them. And maybe in the future, what we can do is have a drug delivery vehicle that stays closed, okay, has inside it the drug, goes and travels to the cancer cell. In the cancer cell, it will, or in the cell, basically, if it encounters these gene products that are biomarkers of cancer, it releases the cargo. It will not release the cargo unless it sees these biomarkers. Now, that would be a very, very useful molecule. Now, we heard a lot of really beautiful talks, including just now, a talk about bioresponsive materials, but this would be gene-responsive materials, basically. Okay, so the last challenge I want to explain to you as well is this area of biologics. So some of you are familiar with the fact that now, other than small molecule drugs, what we have are these biological molecules that are drugs. So essentially, an antibody is a drug. And you saw in the last 2 days some of the power of these biologics drugs because they're very, very specific. Another class of biologic drugs are DNA, RNA, nucleic acid therapeutics. They are really emerging on the market. The 2018 saw the first FDA approval of the first siRNA drug. Okay, 2019 there was a second one, and I can guarantee that we're going to continue on seeing nucleic acid therapeutics going into the clinic and going basically to the patients essentially. What they do is really interesting because rather than targeting the protein itself, the protein is what we target when we have a small molecule usually, okay? They target the RNA or the DNA that gave rise to the protein, okay? And that's really interesting because it's much easier in terms of designing a compound to target the DNA or the RNA. And so there's a lot of modalities, but The reason why you haven't seen them, they've been discovered a while back, the reason why you haven't seen them on the market and they're just now starting to emerge is that there are a lot of barriers to their delivery. So one of the things that is problematic is the low biological stability of these. So you put them in circulation, they're actually not very stable. We'll talk about that in a little bit. Once you put them in and they go to cells, usually a DNA or an RNA is very difficult to enter into cells. It is a negatively charged molecule. The cell membrane is quite hydrophobic. It really has a hard time going in. So that's not good either. Sometimes they cause an immune response, although right now people are saying, well, let's actually use them for immunotherapy. So there's another angle to this. And also, very importantly, if you put them in circulation, they'll probably go either to the kidney or to the liver, okay? And that's great if you want to cure liver diseases, which is exactly what this siRNA that was approved, it is basically for the liver. But if you want to cure other diseases, you need to find a way to take them to the right organ. So a lot of different challenges that we need to address. So let's see what DNA nanostructures can do for us. Okay, let me start by telling you that DNA is the most predictable and programmable molecule that we know right now. And I'm actually saying it is more predictable and programmable than proteins, than RNA, than small molecules. And the reason for that is because it's very simple. It's basically a polymer with a sequence of DNA bases, ATGC, and it is that sequence that determines the full behavior of a DNA strand. And so if a DNA strand finds its complementary partner, well, basically they'll come together into this double helix that we all know. And the double helix itself, from the standpoint of biology, is very, very well defined. It behaves like a rigid molecule over about 10 nanometers. That's actually a pretty good distance. And it has a persistence length of about 50 nanometers. So it's a well-defined object, okay? And so we can maybe start building from DNA. And just to tell you as well, now we don't have to get biological sources to create DNA. We can actually make the DNA in our own laboratory. So in my lab, there are two machines like this one here. They're called DNA synthesizers, and then on a daily basis, my students will basically put the bottles on the machine, go to the computer. It's a newer computer than this one, I think. Go to the computer and basically put the sequence. Just put ATGC, whatever, and the machine by itself in an automated way makes the DNA strand. In a few hours, you have the DNA strand. That's really powerful. What's also really powerful, that allows you to chemically change the DNA, because we need to think about that for various applications. And so the question that was asked now a few years ago is, can we use DNA as a molecular Lego building block? Or probably, some of you probably know this, but very few of you. Most of you played with Legos, but this is an older version of Legos called Tinker Toys. And Tinker Toys are probably more like what the DNA construction strategy that we're going to have today. Okay, so can we do that, and how do we do that? So what I'm going to do is I'm actually going to walk you through how we design and how we make these DNA structures, okay? All right, so we are going to try to see if these guys basically help all of the problems that I've talked about before. Okay, so as I told you, the field of DNA nanotechnology is a young field, and a lot of the approaches in DNA nanotechnology require a very large number of DNA strands. And so, for example, this is the quintessential smiling face, yeah, something like this, smiley face that was made by Paul Rothemund from DNA. Okay, so it's a DNA-based smiley face. It requires 150 different strands and a big, long strand to fold in order to make that smiley face. Now, that would be very interesting for some applications, but certainly for in vivo applications, that's both expensive, and then you have to know the biological properties of all of the 150 different strands that you would be using. So really, for biology, what we need is a simplified version of these systems, and we need something that is not going to lose its functionality. So it has the same functionality, but it's a simplified version. is formed from very few DNA strands. Okay, so we were very lucky to have been at the almost at the very beginning of the field of DNA nanotechnology. And so then at that point, we proposed a different strategy for making DNA nanostructures. And I'm going to try to explain to you the strategy today. But in general, this strategy brings together two fields. It brings together the field of molecular biology and making DNA, et cetera, and the field of chemistry, and specifically supramolecular chemistry. And it says, if I use supramolecular chemistry principles, I might be able to simplify the construction of DNA structures and make them such that they can actually be approved one day by the FDA and applied to different modalities, therapeutic modalities. Okay, so let's start with this. I'm going to walk you through a number of different classes of structures that we have generated. So the first one is DNA cages. Then we're going to talk about DNA nanotubes. And then what we're going to do is very quickly talk about these two new classes of molecules that we've developed. And then after that, we'll talk about their biological activity. Okay, let's talk about DNA cages. Over the past few years, we've proposed a number of ways to make them, but I'm just going to focus on one of them because I really would like to actually walk you through it. All right, so here is a DNA. So this is very difficult because this is kind of hard to control. Okay, there we go. This is a DNA strand. You know, you make it from a DNA synthesizer, or you can call a company, order it, and it will come to you, okay? What we do is we design this part of the strand and this part of the strand to be complementary to the backside of the green strand, such that when these two come together, the brown strand is going to close on the green strand to make a cycle. All right, so I want to make a cube. What do? I design the next one to be complementary to the green strand. Then I design a fourth clip to be complementary, okay? But then at this point in time, this here and this here, we'll design them to have complementary sequences so that this structure actually can close on itself and make a DNA cube. Notice that when you make the DNA cube, what you can have is 1, 2, 3, 4, 5, 6, 7, 8, basically, single-stranded portions on binding sites, basically. Each one of them is a different sequence, so I've got a cube now on the nanoscale, and I can put 8 different things exactly where I want them to go on that cube, and that's really what the power of the approach is. So this is shown just schematically in this movie. We have clip-by-clip approach where these are coming together, they close up onto each other, and then 8 things can come and decorate them in exactly the right geometry for whatever application that you have. And now we're not making them from 150 strands. In fact, we're making a cube from 4 strands. Okay, these are the cubes. Let's talk about nanotubes. Why nanotubes? Well, it's because over the last few years, it has emerged, as I told you, that non-spherical nanostructures, particularly structures that are elongated, are actually very interesting for drug delivery. They have very different biodistributions and different ways of cellular entry, et cetera. So we were very intrigued by trying to make these nanotubes. Okay, again, I'm going to walk you through the synthesis of the nanotube. So I'm going to think of the nanotube here with triangles as basically a bunch of DNA triangles sitting on top of each other and polymerized together. So they're kind of basically coming together, hooked together by DNA strands. So first I have to make these DNA triangles, and then I have to find a way to actually bring them together. Okay, so the way that I do this is I start with a DNA strand shown right here, okay? And I bring another strand, and the way that it comes together with this first strand on these two sides here tweezes, if you want, or closes the first strand into a triangle. So when they come together, basically, the second strand makes the triangle. Then this red strand here fills the third part of the triangle. Now we want this triangle to have DNA strands that stand up and down so that we can polymerize it, right? But single-strand the DNA is very flexible. So what we're going to do is we're going to rigidify single-stranded DNA partially into double-stranded DNA. So we're going to basically place these guys here on so that we can rigidify it, but leaving some single-stranded DNA so that they can come together. So you can see that more, probably easier in a movie. Here's your first strand, comes together with the second strand, and basically the second closes the first. Green strand comes in, we have 3 yellow strands that are partially complementary and that keep everything going up and down. Okay, and now what you can do is basically line them up and polymerize them using DNA strands that are going to come and link them together. Okay, so this is how you do it. There is a little bit of an art to this because you have to do the first step before the second 2 steps so that you can avoid cross-linking. But in the end, what you get are these really nice DNA nanotubes shown right here like needles. And what's really cool is you can choose the DNA strands here to be double-stranded or the DNA strands to be single-stranded. In double-stranded form, the whole nanotube is very rigid and it's closed. And we discovered that in single-stranded form, it's open. Okay, so we can start accomplishing a few things that are really interesting. Before I talk about that, though, I also want to tell you that we've put together methods to control the length of DNA nanotubes and every single level on that DNA nanotube. Again, we had to learn how to do it with the minimum number of DNA strands. Okay, so our nanotubes shown right here are shown before, have very large pore sizes. So in the first generation, what we had to do is find a guest to put inside that is large. It cannot be small, but I'll tell you in a second how we actually solved this problem. So we decided to choose a gold nanoparticle for this process. And so what we're going to do is basically make the nanotube in the presence of the gold particle and encapsulate the gold nanoparticle on the inside, just as a model system, because the idea then is we want a gene-responsive product that closes on a guest and then only opens it in the presence of a gene or a gene product. Okay, so this is what it looks like. Basically, the gold nanoparticles are aligned like peas in a pod, and then you can actually come in and, I don't know if I have time right now. I guess the speaker can't see the time. So, in any case, I might have to skip this. I just wanted to walk you through strand displacement. But basically, what we did is we then added a DNA strand. Okay, that DNA strand, as you're going to see in the movie, is going to make this nanotube, is going to peel off, remove some of the strands from the nanotube. The nanotube becomes single-stranded, and the cargo goes right out. Okay, so this is what it looks like. These are the nanotubes containing cargo on the inside, and now we're going to add DNA strands. They're going to remove the cargo and eject it to the outside, and that happens very quickly, actually. In a few minutes, you eject completely your cargo. Now, of course, this is a model system, but one can imagine that the cargo is a drug, and the DNA strand here is instead a microRNA or an mRNA that actually causes this release. Okay, so 2 more classes of molecules, and then we talk about biology. This is very quickly, because this is kind of like more for chemists, but in any case, I'm going to show you this because it's kind of fun. What we made here are sequence-controlled polymers. And so for those of you, probably most of you know what a polymer is or a plastic is. Usually a polymer is made of multiple identical monomers sitting one next to the other, okay? But natural systems have not chosen to do this for their polymers. Nature has chosen to make polymers in a sequence-defined way. And so, for example, a DNA would have a certain sequence of bases down its length, or a certain protein would have amino acid sequences. And that's because these guys contain a lot of information and can assemble into a very large number of structures. So the question is, as a chemist, how do we create sequence-controlled polymers if, the normal way is going to give you identical building blocks? Well, I told you about the DNA synthesizer. It's an amazing machine. We didn't invent it. 50 years went of invention and innovation went into this machine, and it has revolutionized, basically, molecular biology. What this machine relies on is basically taking a molecule and then just adding building blocks one after the other onto that molecule. That's what it does, okay? The building blocks are usually DNA building blocks. So all we did is we said, well, what about we fool the DNA synthesizer? And instead of using DNA building blocks here, we actually are going to replace them with totally artificial building blocks, but we keep the same chemistry. We don't change that. And so you can go crazy, right? You can have pi rings, you can have hydrophobic units, peg chains, fluorophilic, you can have amino acids, you can have sugars, you can have whatever you want. And now what you can do is you can put them as bottles in the DNA synthesizer, and you can ask the synthesizer now to make these in a sequence-selective fashion, okay? So from these, you're going to get now sequence-specific polymers. And I'm going to show you some of the applications of these, a couple of applications of these in a little bit. Just one thing about these that is very cool is that now what happens is if you have a DNA strand, okay, and you build basically this polymer off of it, this polymer has exactly 12 C12 chains, so basically quite hydrophobic. What happens is, many of you probably would be familiar with this, what happens is if you put this in water, the DNA wants to be on the outside, and the alkyl chains want to be on the inside, and so many of these molecules are going to come together into what we call a spherical nucleic acid. So it's a sphere, basically, with the outside being hydrophilic and the inside being hydrophobic. What's really beautiful about this is that this is a single molecule, a single kind of molecule that has assembled into this. So very, very good for applications. But because we started with a DNA synthesized, kind of DNA synthesized or synthesized molecule, these are monodispersed molecules. They're not mixtures of products. And so the spheres are also monodispersed. They're not mixtures of spheres. Okay, and the beauty of this is just like in natural systems, just like with proteins, you change one residue and the whole structure changes. I don't have time to tell you about this, but it's like a wonderland of structures. So in this case, all we had to do is basically add a single aromatic molecule like this here. This is actually a molecule that emits light, basically, is fluorescent. We added it on the end, and lo and behold, now we don't get spheres anymore. We actually get fibers. Okay, and of course we had to understand why they formed fibers in order to apply this, and now we can actually even control the length of these fibers. Okay, very good. So that's one class, and the last class of materials that I'm going to tell you about are printed materials. For those of you who are interested in other materials right now, so some of you might actually be working with other materials, this might be interesting for you. Okay, just in parenthesis, how expensive is DNA? That's the question that I'm always asked. How expensive is DNA? I mean, a DNA nanostructure, how much is it going to cost? Is it possible to actually make kilogram quantities of it? The short answer is it's definitely possible to make kilogram quantities of it, because now we have kilogram quantities of nucleic acid therapeutics on the market. So that's definitely the case. But how expensive? Well, if I wanted to make this cube, for example, this cube would cost me $100 for 20 milligrams. That's very little and quite expensive. It's going down, but it's not ideal in terms of cost. Of course, antibodies would cost you much more, right? Okay, so that's the cost, but more importantly, a lot of times really what you want is you want to use another chemistry for your material that you're using for drug delivery. You don't really want to use DNA to make the whole material, but you want the information from DNA to be there, like all of the geometrical information from DNA. So what we did is we basically developed a technique where the DNA nanostructure is not used itself as a scaffold, it's used as a printing press. So imagine that you have a stamp that is the DNA, and you bring in other materials and you stamp the pattern of the DNA onto other materials. And you can do this over and over again, basically, and you reuse your DNA structures. And so I'm not going to go into the chemistry of this, but in the end, what you can make are these materials that are not at all DNA, but they have the same information that the DNA contained. Essentially, an organization of the DNA strands on them that allows you to accomplish all of the complex functions of a DNA nanostructures. So here's, for example, a gold nanoparticle, which has basically a certain number of DNA strands in precise ways, and then you can see how you can make bear's paws from it and all sorts of things that are actually quite interesting optically. Okay, and we have actually generalize this to not just gold nanoparticles, but also polymers. Now we can print on polymers. Now we can mold polymers inside DNA cages, so we can make a polymer with the same shape as a DNA cage. And finally, we can also print on DNA strands, on molecules, small molecules DNA strands. So this is a general method, and now we're kind of working towards little microfluidics machines, basically, that labs can buy for cheap, and that allow them basically to print DNA strands on their own materials using our technology. Okay, so now you know how we make our materials, and I'm going to start talking to you about the biological properties. And there's a lot of questions. So this field, as I told you, DNA nanotechnology is very young. It's very, very promising. I hope you've seen how well we can actually control materials with DNA. So it's a very promising field, but we are at the very beginning of the field. So there's a lot of questions that we have to answer before the field can actually take off, biologically at least. Okay? So first question, are DNA nanostructures susceptible to degradation? So if I take a DNA strand and I inject it in the body, after a few minutes, it actually gets completely degraded. We have enzymes in our blood called nucleases that are actually capable of degrading DNA into little pieces. And the fundamental question that we asked is, could it be possibly that if I take DNA strands and I make them into these cages that are very, very compact and closed, could it be that we can slow down this enzyme degradation? So the enzyme basically doesn't know where it can grab onto the DNA in order to degrade it. And indeed, that was the case. So a DNA strand might last, for example, 4 hours in serum, in blood, okay? If you just make the 3 DNA strands into a prism, just 3 into a prism, that's it, then you have an increase of the lifetime to 18 hours. If you just now place on the extremities 2 little polyethylene glycol chains, now we have very respectable lifetimes in serum. We have lifetimes that are about 60 hours. So this is good. It is very interesting, begs the question, why does the enzyme actually not recognize it? And there are all these really interesting fundamental questions we need to answer for this. So that's good. Let's talk about the next one. I told you that the problem with DNA therapeutics or RNA therapeutics is that they're not taken up by cells. Could it be, again, that our shapes are recognized differently by receptors on the surface of cells? Okay? Could it be that there is a receptor that actually recognizes our stuff differently and then brings it in? Is it possible? Okay? And here's where the problem is really interesting, has amazingly good results, but also I want to tell you about some of the other results, and I want to give you a word of warning here. Okay, so do they penetrate cells? And the question is, if I have all these beautiful shapes from DNA, will they be recognized by some kind of receptor and be brought into cells? Okay, well, the first thing to say here is that if you have a spherical nucleic acid, the one that I described to you before, the mice cell, that is avidly taken into all sorts of mammalian cells. Sick cells, healthy cells, it goes into skin cells. We worked with L'Oreal, actually, to deliver these in the skin. So it's a beautiful modality. It works really well. It goes in. Again, begs the question, what is the receptor that is actually bringing it in? Very, very interesting questions that we're working on. However, there is something else that we ran into when we started working with full DNA nanostructures, not ones that were hybrid structures, but full DNA nanostructures. In the literature, and in many of the DNA nanotechnology conference, there have been some papers in 2010 that have said DNA nanostructures, like the 3D cages, enter cells very well, okay? And then it has become a folklore to say, now, DNA nanostructures all enter cells very well. Let's go to the next phase. Definitely they enter cells. That's basically the mantra, okay? However, the experiments that were tried, that were used to actually show this, have some problems, and I'm going to explain them to you. The problem is when you do an experiment to see if something goes into a cell, what you do is you take a DNA structure, you place on it a fluorescent molecule, so a molecule that emits light, okay? And then you put it with cells, you incubate it, and you look inside and see if there's fluorescence on the inside of cells. And indeed, wow, there's a lot of fluorescence. In fact, if some of you might know biology a little bit more, cell biology a little bit more, you might say, oh, this is mitochondrial. Wow, we're delivering to the mitochondria. That's fantastic. No, that's not the case. Through a lot of control experiments and a lot of hard times for my beautiful student Aurélie, we were able to show that, unfortunately, many of these studies are flawed because what's happening is you are incubating, these nanostructures are being degraded by the enzymes in the serum into tiny little pieces that contain the fluorescent molecule. And the fluorescent molecule itself, of course, knows how to travel very nicely into the cell. In fact, it loves to go to the mitochondria. You are not delivering your cages, actually. You're delivering the small molecule attached to the cages. And I want to really, really caution any of you guys, students that are working in cell biology about this. Control experiments, as many as you can, okay? You can have so many artifacts, especially from fluorescence microscopy, and you really need to be very careful. What is the bottom line? The bottom line is DNA nanostructures actually do enter cells. You can see they do enter cells, but not to the same extent as what people think. And they're not avidly taken up by cells, not all of them, and it's very cell-specific, it's very structure-specific, and one has to actually think about that before starting to do work with cells. Okay. Now, they don't enter cells by themselves, or they enter very little. I'll tell you how people make them enter cells. Question is, therapeutic activity. I told you that we want to use nucleic acids as therapeutics. They silence gene expression. They basically are fantastic therapies. But let's say we place our DNA therapeutics on a cage, on a DNA cage. Will it still be biologically active? We don't want to lose the activity of the structure by placing it on a DNA cage, right? And the answer is, not only is it still active, it's much more active than the DNA strand by itself. We don't know exactly why it's much more active. We just know that the DNA cage increases the resistance of this to nucleases. Probably that plays a role in why this is very active, but it's great news. And this is actually a general situation. Spherical nucleic acids are like that. We have other hybrid structures that are like that. These DNA nanostructures are actually very potent for gene silencing, which is fantastic. But you can see the molecule on the bottom here. For these studies, we did not use naked DNA structures like I showed you before. We had to use a positively charged a polymer or a positive charged lipid in order to deliver them into cells, okay? And that, to me, is going to be the next challenge. How do we deliver DNA structures naked without the need to use these positively charged polymers or lipids? They're not the end of the world. In fact, one of the siRNAs that is now on the market is delivered through a positively charged lipid, so it's not that bad, but it would be really nice to find mechanisms, cellular mechanisms, whereby you can deliver this without the need of anything. Okay, they go into cells. Let's actually accomplish that thing that we talked about from before. We talked about gene delivery, right? We talked about creating products that respond to specific genes. Okay, what we're going to do now, since we know that they enter cells aided, but they do enter cells, what we're going to do is we're going to make a cage. The cage is closed, and this cage is designed so that it responds to a gene product, so basically an RNA strand. This RNA strand is only expressed in prostate cancer cells. It's not expressed in normal cells, okay? And we're going to, for those of you who know molecular biology, it happens to be a fusion of two genes that are in normal cells. So it's a fusion product, and we're going to design a cage that opens only with this product. And the mechanism is shown right here. What this product is going to come and do is it's going to open one side, and as you can see, you need two molecules to open the two sides. So you can actually maybe have two different markers for this, okay? And then open this. If you have cargo on the inside, the cargo is going to be released or at least revealed, which is really nice. What cargo are we going to, did we do anything, do we put cargo? Yes, we did. So Catherine here made a molecule she called the suitcase, the nano suitcase prism. So it's a suitcase because it's carrying on the inside an siRNA, okay? And the siRNA is completely inactive. So you take this into cells, it's completely inactive, but in the presence of the correct microRNA, it actually gets released and it achieves silencing. So now we have gene product responsive DNA cages from very, very low number, basically, of DNA strands. Okay, two more questions. I told you in the beginning that we had to encapsulate these gigantic gold nanoparticles inside our cages, and that's somewhat useful, but not very useful because most drugs are actually small molecules you want to encapsulate on the inside, right? So how do we make these cages encapsulate small molecules without the small molecules just coming out, essentially? Well, we ran into this completely by accident. Okay, so Tom here, what he wanted to do is basically create these DNA strands that have lipids on the exterior. They have a patch of lipid on the exterior. And the reason why we wanted to do that is basically with the lipids on the exterior, we would facilitate the insertion of these into cells. That's really the beginning of this. However, what we discovered is the following. If you have precisely 8 lipids patches on our DNA cube, okay, the lipids do not go outside anymore. They all go inside, and they shake hands on the inside of the cage. And they make this little cavity here of hydrophobic stuff on the inside right here. So this is a very easy way to take a cage and to make a hydrophobic pocket on the inside, a bit like an enzyme pocket, a hydrophobic pocket on the inside in order to encapsulate small molecule guests. And we teamed up with Jianing Li and basically her student Cheng Yi, who's at University of Vermont, and she did some molecular dynamic simulation of that handcake, and you're going to see it right here. The blue strands here, the blue things here are the hydrophobic chains, and as you can see, you let them go, and they actually kind of cluster with each other, and they basically make these kinds of dynamic pockets on the inside. And indeed, now what you can do is you can put in small molecules, and doing the same thing that I showed you with the gold nanoparticles, you can add a gene product, and you can actually release these from the inside. Okay, so that's really good. Last, do they have any interesting in vivo behavior? Okay, so this is, I'm just going to give you one example. We've been doing a number of in vivo work, vivo studies, but I'll show you this one. So this is a collaboration with Larry Panache. Larry is a medical doctor, an oncologist at the Montreal Jewish General Hospital, and he is basically interested in many patients, including colon cancer patients and other patients like this. And he's also interested in chronic lymphocytic leukemia patients, CLL patients. Okay, so these are leukemia patients, and sometimes what happens is these patients develop resistance to chemotherapy. And there is a drug out there. This is the drug. It's called WEP2020. It has been discovered, and it is well known to actually resensitize cancer cells to chemotherapy. That's nice, okay? So why don't we use this drug? This drug is stuck in clinical trials, and it's just not making it out of clinical trials. Why? Because this drug actually has a lot of side effects, including one where it actually crosses the blood-brain barrier and causes severe depressive episodes. And so you can imagine a cancer patient, of course, with all of the other comorbidities. In addition to that, you're giving that patient high amounts of depression, essentially, by doing this. So I don't know if it's going to be likely to make it past some clinical trials. So we had a very, very simple question. Can we put that drug on the inside of, let's say, our spherical nucleic acids, and can we actually bypass this problem? The spherical nucleic acids would carry it, and perhaps they would not distribute to the brain. Okay? And also, this will allow us to ask the question, where do they go? Do they all go to the liver, or do they go to other places? Okay, so to cut a long story short, we were able to stably encapsulate these and indeed show that, that even when they're encapsulated and delivered to cells, including primary cells from CLL patients, they actually can resensitize cells to chemotherapy. So that's all good. But now, what do they do in vivo? So I'll show you just a little bit. This is intraperitoneal injection in CDR mice, and hopefully you can see this. So what you're looking at here are little colored things, and you're going to start seeing organs. And you can see that the colored green, yellow stuff is where our molecule is, our spherical nucleic acid with. This is the liver. You can see it's not all localized in the liver. It's just basically everywhere in the body at this point in time. Okay? Now it's going to go to the kidneys. I don't know if you can see them. Can you see the kidneys? I don't know if you can see anything. But basically, these are the kidneys, and it's not really localized there. You cannot see very much those green. It's there, but it's not a lot. Okay, it's not solely localized there. I think it's going to go to the lungs. There's a little bit in the lungs. You can see it right here. But it's, again, not very, very much localized there. So that's good. It distributes well. And most importantly, it does not cross the blood-brain barrier. And it does not go to the brain, and it will not, at least in this case, it will not give us these side effects. Interestingly as well, these actually are able to well, they last much, much longer, actually, in organisms than normal nucleic acids. But very importantly as well, they localize in tumors. And we heard some nice talks about the mechanism of localization in tumors and whether it's APR or transcytosis. It's a very interesting discussion, but they seem to be also localized. So they do have some promising in vivo properties, but we have a long way to go before really validating these, and we're at the beginning of this road in terms of in vivo work. So I hope that I have been able to show you today that you can make DNA nanostructures with precision that is not achievable with other nanomaterials. And for us, that's really, really exciting because they are going to enable a whole variety of precision medicine that was not there before, including things like gene product response and presentation of ligands to target specific receptors, et cetera. There's a lot of application. I'll go over them. So what I hope to have shown you today is that in some cases you have enhanced cellular uptake. The red stuff is the question marks, what we have to really understand right now. They actually can achieve gene silencing very well. Okay, they have a favorable in vivo distribution in some cases. They can release very selectively their cargo with cancer-specific signals, including gene product, which is really nice. And they allow you to actually start exploring systematic structure activity studies. What is the effect of structure on drug delivery? And the big question, well, the big question is, can they enter cells in a naked form? How do we do that? The second question, though, is, can we engineer them to not all go just to the liver or the kidney? You know, to go to other organs so we can address other diseases. And finally, we can place ligands exactly where we want them to go on these structures, and therefore we can start examining things that we were not capable of examining before, which is, for example, if I have a PD-L1 receptor, does it actually have some kind of organization on a cellular surface? We heard about the trimeric PD-L1 receptors that you developed, Leaf. You know, could we actually change the geometry of these PD-L1 ligands and actually accomplish different kinds of biological activity? This is just the beginning of the journey in terms of this. I have not been able to tell you about the interaction of our cages with the lipid bilayer, but just to cut a long story short, they're amazing. If you put cholesterols on them, they can either float on the outside of cells or lipids, or they can actually, if you put them on the cholesterols on the two sides, they can actually dip themselves inside the membrane, okay? And they can create nanopores, which is really surprising given the fact that the DNA itself is negatively charged. It's embedding in a lipid membrane. So we also have a long way to go here to understand this, but it's very, very exciting. Okay, and it remains for me to thank my wonderful students for this work. Before I forget, though, I do want to say something, because it might actually be a question in your head, and I forgot to say it at the time I wanted to say it. You see this beautiful DNA structure? I mean, I have to commend the organizers in every which way. It's amazing. And so it's kind of fun to find a single flaw in this WEP organization. It's so perfect. There is one flaw, just one. Okay? And the single flaw is that this DNA that they're showing right here is left-handed. It's not right-handed. So right-handed DNA is our DNA in our body. But this is really cool, because it's actually allowed me to tell you that a lot of people worry that DNA nanostructures are, well, they're made of DNA. What are they going to do in our body? Could they actually start hijacking our own DNA and doing weird things to our DNA, right? No, because what we're going to do is we're going to build them from other kinds of DNA, including we're going to make them from left-handed DNA. There's a lot of new research now coming out to make left-handed DNA. It behaves exactly like right-handed DNA in terms of how it assembles, but nothing in your body recognizes it. Nothing, okay? And it's not toxic. So it's fantastic, right? And in fact, it actually resists nucleases a little bit better, too. So thank you to the WEP organizers and the WEP visualization people for allowing me to actually also show you this. And thank you so much for your attention.
Okay, thank you. I think all of you agree with me that this is probably one of the best talks that we had so far. Thank you so much, Hanadi. And just to comment on the DNA, this is the futuristic approach. This is the left-handed DNA.
Absolutely.
Well, actually, I had no clue, but now I know. You told me about it. I'm a chemist. I just know molecules, but thank you. Now we know. But it's a futuristic approach. I'm happy with it. So what about some questions? Yes, Leaf. Can you take this? I just want to see. I'm just worried. You traveled today? just in case. Well, I'm going to try it. Yeah, let's go.
Okay, I got it. Wonderful. I thought I'm going to fumble. So obviously, your DNA nanostructure, after getting to the endosome, they can escape. What is the mechanism of endosome escape?
Okay, so what we have seen right now is only escape when we add a lipofectamine or we add, so they are not able to escape. Now, there is word in the literature. If you go into the DNA nanotech literature, you will see, for example, people using nanotubes and saying that the aspect ratio of a nanotube allows them to escape endosomes. Frankly speaking, right now, I'm a little bit skeptical of everything that's coming out because of our cautionary tale. I think we have to do it ourselves, or other people have to do it with a ton of control experiments to see if we can use shape to basically get us out of the endosome. But that's definitely an area of research.
Okay, so regarding the left-handed DNA, although the left-hand DNA is not toxic, but if they're not degradable, they're going to stay in the body for a long time. So
That's
for
true.
a short time, there's not toxicity, but long time, you've got to be very careful.
Yes, yeah, you're absolutely right. I think that what we might have to do is figure out a mechanism whereby you can actually somehow cleave it, but much, much later. Like, you know, to have essentially a cleavage mechanism so it can go to small molecules, but that it's not going to remain in circulation. There are other approaches, by the way, right? You can use phosphorothioates, for example. You can use phosphorothioates. You can use FANA, so fluorinated nucleic acids. There are many other approaches for this. I just wanted to use this as an example. But and these actually don't stay in circulation.
Okay, other questions? Right there. So Tahani, it's up to you. You can throw it. Yeah, go ahead.
I don't know. I'm not going to throw it.
Okay, that's fine. That's fine. Halfway through, just okay.
Thank you so much for the very wonderful talk. We have this big debate going on nowadays in the world about healthcare cost. And I was just wondering, in that context, where do you think the chemistry and biology you presented today would take that healthcare cost towards? Would it be more expensive medicine, or would it be less expensive medicine which is affordable for all?
Yeah.
Thank you.
Yeah, this is something that really occupies us very much. And you can see actually that from the very outset of my research, I wanted to make DNA minimal structures to reduce the cost. And then I even moved to hybrid structures to further reduce the cost. And then I moved, in the end, to printed structures to further reduce the cost. So we're trying to work at these strategies to actually get this to be available. But currently, if you go to, for example, the last siRNA-approved drug, patisiran, I think it's called, it's hundreds of thousands of dollars. And you know what? I don't know. I mean, they're trying to pay for the research that they did, right? Because it does not cost hundreds of thousands of dollars to make. You can come to my lab and make it for much less. So, but of course, you have to understand that this is drug discovery, so it takes a lot of money for that. Once it goes off patent, though, actually, it's not going to be that expensive. So it's getting lower and lower, the cost of making DNA. DNA synthesizers are getting bigger and bigger. We're now using, rather than solid phase and vacuum and stuff like that, we're using flow chemistry to do this. So chemists like Naveen are actually solving some of these problems.
Yeah, and just to comment, carbon nanotubes, when they started, everything starts very expensive. But when things are working, scale up would find a way to make it cheap. Carbon nanotubes, when it started, it was like $1,000 in graphene and graphite. Now it's like you can buy tons.
Yeah.
So yeah, costs will definitely be better. One last question, or we have more than, okay. So we'll start here and then we'll go over there. So 2 last questions.
Thank you for
I'm
this.
not a good thrower, so.
Thank you for this talk. I have a question. You mentioned that the complexity of DNA nanostructures is a problem for FDA approval. There are approaches where they use click chemistry or this UV dwelling to connect the staple strands and to generate, ideally, just 2 molecules. Do you think this is a viable approach?
I think this might be a really interesting way to get around the problem of the FDA thinking of it as a mixture of DNA strands. But it doesn't get over the problem that no matter what, your 200 strands are going to be super expensive to make. Now, one day, if the DNA synthesis cost is going to go super low, maybe that's going to be the way. But, I think philosophically in my head, if you can achieve the same exact function using a DNA minimal structure, why would you go and make a smiley face? I mean, you can make it for something very interesting, but why would you do that if you can actually do the same thing with a wireframe DNA structure? That's just my opinion.
So question here. So very interesting talk.
Yes.
But, concerning the left-handedness and the right-handedness, you also said during the talk that, it needs to interact with microRNAs and other structures. So is that then not a contradiction?
No, it's not, because what you do, for example, is you construct the whole thing from left-handed DNA or some other DNA analog. It doesn't have to be left-handed, but a DNA analog that is not actually recognized, okay? And then what you do is you make the strand. So you make the strand that hybridizes with the cage, let's say. Half of it is left-handed, and half of it is right-handed. You could do that very easily by DNA synthesis on the synthesizer, right? So then the whole thing is left-handed, is not going to be chewed up by anything, but the half that's right-handed is actually what you're going to be using as a therapeutic. So that's basically how you get around it. Again, chemistry is what. It's all about the chemistry. Well, actually, it's both biology and chemistry.
Yes. My question is about the degradation. You showed.
I'm sorry, I don't see. Hi. Okay.
You showed how it's possible to make these structures survive for, let's say, 60 hours. And you also showed how you can open them up to release them in the correct places. But let's say after 62 hours, all of them will open up no matter at what place they are, right? How do you work this out?
Yes, a very, very good idea, a very good point. So I didn't have time to actually discuss this. There are two modalities. If you're talking about siRNA release modalities, what we're hoping to do is to regulate the kinetics of all of this. So you've got the siRNA on the inside, you've got the opening and the release. We want the release to be fast when there is the microRNA, let's say, okay? But then if there is no microRNA, we want the degradation of siRNA to then kick in. Do you see what I'm saying? Because it will degrade, under the right conditions. So by regulating the kinetics, we can do this. For small molecules, you're absolutely right. You cannot take those small molecules and bring them into cells and expect them to be basically. So for those small molecules, what we're working with are these things called aptamers. So they are basically nucleic acids that recognize proteins on the outside of the cell. Because on the outside of a cell, you can do that kind of a conditional release at that point. Okay?
Yeah. Hi, thank you very much for the talk. I want to preamble this by saying I failed Biology 101, so that's why I went into security. And my question is related to security.
So I just wanted to say something for everybody in the room. This is Ed Sleiman that sends you the phishing email.
Sorry. Right, exactly. So from a security standpoint, so once you get into the in vivo aspect of this, can this be dangerous? Can it be used if someone injects someone with something that's malicious? Can this harm someone?
I would say that if you're using DNA analogs, like not DNA itself, but some analog that is not going to cause itself sort of a nucleic acid thing, I would say not more than a normal drug. I mean, you can inject a person on the street as well, a small molecule drug, and also cause the same thing. So no more than that, for sure. There are very smart people out there that might actually use it in a different way for, But it isn't a technology that intrinsically opens itself up to more security problems. Because it is chemistry, it's not a biological molecule that we're using.
But since you do this on a computer, so a programmer can program something that's malicious and then also, that's what I meant.
I see what you're saying. So good point. Good point. That's definitely something that we have to worry about. Yes, that a lot of DNA design is actually made computationally. Basically, we have algorithms for this. Now, just to say, our algorithms are super, super easy because we have so few strands. But yes, if you're trying to make a 3D DNA origami or a Stanford bunny, you saw the bunny, the rabbit. If you're trying to make a rabbit, yeah, you definitely need computational tools for this.
Okay.
Yes.
Thank you.
Okay, let's thank our speaker for a great talk. And a quick note for our students, we do have a session about publishing. Hanadi is an associate editor in JAX, and we have Professor Andre Nall from ACS Nano. So how to publish good papers and high impact journals at 2 p.m. today. You can check your schedules. Thank you so much.